Hi, I'm Anupama Jha. I'm a senior CIS PhD student with Yusuf Parash at the University of Pennsylvania. Today, I'll talk about RBP Pokedex. It is an unsupervised deep learning approach for predicting the effect of an RBP's knockdown on other RBPs in healthy human tissues. RNA-binding proteins are major drivers of post-transcriptional regulation in cells. They preside on processes ranging from transcription, splicing, mRNA transport, stability and localization, translation to proteins, as well as mRNA degradation and storage. RBPs form the regulatory layer that is a key determinant of protein levels and thus cell phenotypes. RBPs can bind and control many different RBP targets. They form complex regulatory networks. For example, RBPs can directly regulate their RNA targets or indirectly regulate them through other RBPs. There can be more than one intermediary forming an RBP chain, or indirect regulation can also occur through a common RBP that controls multiple targets through different RBPs. They can regulate targets through different modes of regulation as well. RBPs can bind cooperatively to a target or compete with one another for this binding. They can also regulate targets through mutually managing each other's expression, and some RBPs auto-regulate their expression. Here I'm only showing you regulation in the 3' UTR, but similar regulatory modes exist for gene expression through 5' and for splicing through binding sites flanking an alternative exon. RBP networks can act in a tissue-specific manner. Multiple RNA binding proteins bind to pre-mRNA to control splicing in a tissue-specific manner. Similarly, gene expression is regulated in different tissues through binding to 5' and 3' UTR. However, systematic experimental characterization of effect of RBP knockdowns on gene expression across different tissues is infeasible. Therefore, we instead ask the question, can predictive modeling of pan-tissue gene expression measurements of RBPs capture tissue-specific RBP state? What we want is a model that learns gene expression of RBPs in different tissues. How do we do that? We train a variational autoencoder on gene expression of 531 RBPs in 47 human tissues with approximately 7,000 samples. Input to the model is gene expression of these RBPs in a tissue sample that goes through a variational autoencoder that learns to compress and decompress the input to reconstruct the same gene expression as the output. Let's evaluate the performance of this VAE. First, we look at test samples for the 47 tissues from training. We compare VAE to a baseline. Our baseline is mean gene expression of RBPs in a tissue. On x-axis, we show variance explained by the reconstruction. Higher values are better. And on the y-axis are the 47 tissues. Our VAE outperforms the baseline and is able to learn RBP gene expression across all tissues. Then we move on to unseen tissues. We withhold 2 tissue groups consisting 6 tissues from GTEx during the VAE training and now measure performance of our VAE on these tissues. Our VAE still beats the baseline even though it has never seen these tissues before. So far, we have shown that we can reconstruct RBP gene expression across different tissues. Our next question is, can we simulate the knockdown of RBPs through these predictive models? The first step then is to train another VAE for 2 human cell lines from the ENCODE project. Since we only have 2 cell lines, we initialize this VAE through our learned weights and biases from the GTEx VAE network. Input to the model is gene expression of 531 RBPs from the HEPG2 and K562 cell lines, and output is their reconstruction. The second step is to actually simulate the RBP knockdown. We do that by reducing weights of the outgoing edges from the neuron in the first layer of the VAE that represents the target RBP, and by reducing expression of the target RBP in the input. These steps lead us to the question, why should it actually work? Our hypothesis is that in order to learn reconstruction of RBPs across different tissues, a VAE has to learn the correlation structure between these RBPs, and thus disrupting that structure by reducing edge weights and expression should simulate the effect of an RBP knockdown. To evaluate whether it actually works, we look at the ENCODE project. ENCODE project contains approximately 245 RBP knockdowns each for HEPG2 and K562 cell lines. However, these experiments were processed in batches, so we need to mitigate batch effect. We use Combat to do that. Here are the results of that process. On x-axis are different sources of variation, and on the y-axis we have % total variance explained by that source. First two bars are the RBP target, a desirable source of variation. All others are undesirable sources of variation. Blue represents before and orange represents after batch correction. You can see that batch correction has retained RBP target variation while reducing other unwanted sources of variation like batch and platform. Then we plot the UMAP of differential gene expression for all RBP knockdowns in the HEPG2 cell line. Before batch correction, experiments cluster by batches, and two U2AF1s that were processed in different batches are at the opposite end. After batch correction, the batch clusters are gone and the two U2AF1 move closer together as expected. And we saw similar effects upon batch correction in K562 cell lines. Now that we have our batch corrected experimental knockdowns, we want to evaluate our in silico knockdowns against them. We measure precision and recall of overlap of RBP targets between the experimental and simulated RBP knockdown. Precision is defined as the ratio between true positives found by the model and total number of positives found by the model. Here we compare precision of our VAE model with a PCA reconstruction. PCA is a linear method that generates reconstruction through a linear projection. Variational autoencoder is a nonlinear method that captures nonlinear dependencies and correlations. We see that our VAE offers moderate precision in capturing RBP targets of knockdowns. Then we look at recall between PCA and VAE. Recall is defined as the ratio of true positives found by the model and total possible true positives. Our VAE offers high recall, meaning that it finds most targets of RBP knockdowns. Therefore, our VAE finds targets of unseen experimental RBP knockdowns with moderate precision and high recall. So far, I have showed you that we can reconstruct RBP gene expression in tissues and simulate RBP knockdown in at least 2 cell lines. Next, we follow up on one RBP-RBP interaction found by the model. Our VAE predicts that in HEPG2 cell line, UCHL5 knockdown alters gene expression of HNRNPK, and similarly, HNRNPK knockdown alters UCHL5 gene expression. This suggests that there is potential of some regulatory entanglement between these two. Therefore, we first look at RNA-seq evidence. So we look at HNRNPK expression in RNA-seq of HEPG control cell lines and UCHL5 knockdown. And this expression is significantly altered upon UCHL5 knockdown. And Similar observations were made for UCHL5 upon HNRNPK knockdown. So our first hypothesis is that these two RBPs are directly regulating each other through binding in the 3' UTR. And in line with that, we found E-clip peaks for HNRNPK binding in the 3' UTR of the UCHL5 gene and found E-clip peaks for UCHL5 binding in the 3' UTR of the HNRNPK gene. So next, we want to know if these two RBPs are involved in cooperative or competitive regulation of other targets. We found significant overlap between their knockdown targets, and log2 fold change of these common genes in these two experiments generally agree. Then we looked at clip binding evidence of HNRNPK upon these two knockdowns and find significant differences between binding peaks found in targets of HNRNPK and UCHL5 knockdown versus non-regulated sets. Similar observations were made for UCHL5 clip binding in UCHL5 and HNRNPK knockdown targets. So there is binding of both these RBPs in each other's knockdown targets. To show cooperative regulation by these two RBPs, we show magnitude of altered expression upon either knockdown in direct or indirect targets of these RBPs. Therefore, on x-axis, we show log2 fold change between control and UCHL5 knockdown on left and HNRNPK knockdown on right. And on y-axis, we show CDF showing cumulative frequency of genes till that log2 fold change threshold. Here we can see higher magnitude of altered gene expression in genes bound by both RBPs, shown here in red, when compared to genes bound by UCHL5 only, shown here in blue, or those bound by HNRNPK only, shown here in black. Least expression change is in the genes that are not bound by either. So altogether, these results suggest that there is potentially some cooperative regulation between these two proteins. Additionally, we think that this relationship explains some of the indirect effects of depletion of either proteins individually. That is, genes that change upon UCHL5 knockdown but are bound only by HNRNPK clip and vice versa. This is only one example that we explored, but our model found many more of such potential co-regulators, and we're planning to explore some in the future. In summary, we show that we can reconstruct RBP gene expression using a variational autoencoder in seen and unseen tissues. Using this VAE, we can model the effect of an RBP's knockdown on other RBPs in silico and in an unsupervised manner. Furthermore, we can show that our model finds real RBP targets through systematic evaluation with large-scale RBP knockdown evidence in form of RNA-seq and CLIP from ENCODE, and through individual validation, we show possibly cooperative regulation between UCHL5 and HNRNPK. Finally, we provide RBP-Pokedex as a tool to the RNA community. It can be used to explore RBP interactions across 53 GTEx tissues and generate RBP regulatory hypotheses. With that, I'd like to thank my PhD advisor, Professor Yusuf Barash, my thesis committee, and members of the Biocyphers Lab, specifically Matthew and Caleb, for countless discussions about this tool and potential applications, and Paul for designing the RBP Pokedex website. I am actively looking for postdoc positions at the intersection of machine learning and computational biology. So please contact me if you have such a position available. Thank you for your time and patience, and I hope you enjoyed this talk.